Hello, everyone, and welcome to today's Gen Biotechnology webinar entitled Harnessing RNA Metabolism for Precision Therapeutics. This webinar was made possible through sponsorship from TriLink Biotechnologies. I'll be the host for today's webinar. My name is Fei Lin, Senior Editor of Gen Biotechnology, Gen's sister peer-reviewed journal covering outstanding research and perspectives across the biotech field. We're pleased to welcome our speaker for today's event, Dr. Jeff Coller. Dr. Coller is the Bloomberg Distinguished Professor of RNA Biology and Therapeutics at Johns Hopkins University. His lab has made seminal discoveries in the area of messenger RNA stability and translation. Prior to moving to Johns Hopkins University, Dr. Coller served as director of the RNA Center at Case Western Reserve University, where he held the Henry Wilson Payne Distinguished Professorship. His publications have been cited over 6,000 times, and he currently holds numerous patents for RNA-based therapeutic applications. In this webinar, Dr. Jeff Coller will describe a new therapeutic technique that enhances mRNA translation. This technology has numerous clinical applications and offers key benefits. For example, it is disease-modifying, restoring normal protein levels. It is mutation agnostic. It can be tailored to precisely control expression. And lastly, it is applicable across indications and highly versatile. If you have a question for our speaker, please input your questions in our Q&A box and stay tuned for our live Q&A session at the end of the webinar. With that, let's bring Dr. Jeff Coller to the stage.
All right, well, good morning, everybody. It's a pleasure to be here today to tell you about some of the work from my lab. I'm going to tell you about a few stories on how we're using our knowledge of RNA metabolism to improve human therapeutics for specific diseases. Just to start out, as a conflict of interest statement, I'm a founder of Tevard Biosciences and a member of their SAB and hold a financial interest in their company. All right, so just to review the central dogma, mRNA is, of course, the message that is derived from DNA and transmits genetic information into the cytoplasm, where that information is then read by ribosomes and translated into protein. And one thing that we think about a lot in our lab is how we preserve this linear flow of genetic information from DNA to protein. And a critical aspect of that is, in fact, the degradation of the mRNA. And the reason for this is that if you stopped a signal of DNA transcription in the nucleus, that signal is pretty much irrelevant as long as the mRNA persists in the cytoplasm and is making protein. So the degradation or the regulated timing of degradation of an mRNA is critical, a critical aspect of gene expression, and as I said, maintaining the linear flow of genetic information from DNA to protein. One piece of information that we're going to talk a lot about today is the presence of a poly A tail on the 3' end of an mRNA. The poly A tail is a critical determinant of mRNA expression. If an mRNA has a poly A tail, it is translated, and it's translated robustly. It's maintained in a stable as a stable species. But if that mRNA loses its poly A tail, which all mRNAs will over time, it no longer translates and is typically degraded. And so the poly A tail is a key node in mRNA metabolism. Importantly, the way in which the cell maintains this genetic flow of information from the nucleus to the cytoplasm is by timing the degradation of the mRNA, specifically by timing how fast that poly A tail is removed. So as soon as the mRNA is exposed to the cytoplasm, there are enzymes called deadenylases that are removing the poly A tail at a defined rate, which is specific for each individual mRNA. And as soon as that mRNA loses its poly A tail, as I said, it's either translationally repressed or degraded. And so deadenylation becomes a critical aspect in controlling the linear flow of genetic information. Every mRNA in the human body is degraded at a different rate. Some mRNAs are degraded very quickly. Some mRNAs are very long-lived. And then there's a complete repertoire of mRNA half-lives everywhere in between. And my lab has studied this process for many years to try and understand how we get these discrete rates of mRNA half-life that are specific for individual transcripts. And a key feature that we've studied for a number of years is that we've known since the 1970s, that the translation rate of an mRNA is absolutely proportional to its mRNA decay rate, such that mRNAs that are translated very well are degraded very low. So in fact, these mRNAs are very stable, where an mRNA that's translated poorly has a very high turnover rate. And so there's an intricate relationship between mRNA translation and mRNA stability, such that they're inversely correlated. And it's this relationship between mRNA degradation and mRNA translation that my lab has spent the last 20 years studying. And what we've determined over the last 20 years, essentially, is that the process of mRNA degradation occurs in a stepwise fashion. First, the mRNA is degraded by removing the 3' poly A tail. This then leads to the decapping of the 5' end, the removal of the 5' M7 GPPG, and then the degradation of the mRNA in a 5' to 3' direction. And what we've shown is that this entire process is occurring co-translationally. It's occurring while the mRNA is embedded in polyribosomes. And the process, in fact, has evolved to dance around polyribosomes in order to not interfere with translation, but to slowly limit translation by removing the tail and then removing the cap so that no further translation can occur. And then exonucleotide decay is occurring 5' to 3' as it's following that last translocating ribosome. This seems like a fairly simplistic process, but there are over 30 proteins that are known to be involved in these discrete steps, with a number of proteins associated with the deadenylase, equal number of proteins associated with the decapping complex, and only a single major factor that's involved in degradation of the mRNA. And so it's in fact these proteins, this large cohort of proteins that is providing the regulatory aspects of mRNA decay that gives the discrete half-lives to individual transcripts. What we've also shown is that an understanding of how mRNA half-life is communicated to the mRNA translational apparatus, and this is through a concept called codon optimality. And what we observed a number of years ago was that the speed of the ribosome is in fact what dictates mRNA half-life. So that the faster the ribosome is translating through an mRNA, the more stable that mRNA becomes. Where if the ribosome is moving slowly through the mRNA, that is an unstable transcript. And a major feature that dictates overall speed of ribosomes is in fact the concentration of functional tRNAs. And so the process of decoding is a stochastic one, and the ribosome has to find the right tRNA for each cognate codon present within the A site of the ribosome. And how fast it can find that cognate tRNA is a function of tRNA concentration, such that if the tRNA concentration is high, that's considered an optimal codon, the ribosome will move fast. If the tRNA concentration is low, that's a non-optimal codon, and the ribosome will move slowly. And these events are then communicated to the mRNA degradation complex. This pathway of mRNA decay, of codon optimality mediated mRNA decay, is very powerful and can explain the repertoire of mRNA half-lives that are observed within an organism. So here's an example of an experiment where we take an mRNA and encode it so that we get the exact same polypeptide sequence, but we're going to change the optimality of the codons using synonymous codons that are either non-optimal or optimal, and just showing the experiment in two different ways, just to show that it's not a function of the way in which we monitor mRNA half-life. And so you can see when the mRNA is fully non-optimal, it's very unstable. But as we increase the optimality of the mRNA by 10%, we slowly increase the overall stability of the mRNA, such that by the time that we reach the end of the experiment where we fully optimize the mRNA so that it has 100% optimal codons, we've achieved every observable mRNA half-life within the cell. And importantly, we haven't changed polypeptide sequence. And so just the simple use of synonymous codon alternatives can modulate mRNA stability in a powerful way. And if we do this, Mother Nature certainly has. And I won't go into more details about this at this time because this has all been published in the past. Moving forward, we've identified the mechanism of how the ribosome speed is sensed by the mRNA degradation process. And everything that I'm showing here in orange are proteins that will sense how fast the ribosome is moving. And as you can see, all of these proteins are really components of what is called the CCR4 NOT complex, or the deadenylase complex. Specifically, we've identified that a key player in the deadenylase complex, called C003 or NOT5, depending on the organism, actually finds its way into the ribosomal E site in response to low concentrations of tRNAs, such that the NOT5 or NOT3 will touch the P site tRNA that is embedded within the ribosome and has a communication with this ribosomal protein S25. And essentially what NOT5 and C03 are recognizing is a situation where there is an empty E site. So just going back to your freshman biology class, the way ribosome translation works is that there's an A, a P, and an E site, and the tRNA that has the amino acid that's recognizing the cognate codon present within the A site comes here. And then that transfers the amino acid to the P site, and then now that vacant tRNA goes into the E site and is then ejected from the ribosome. If the process of finding that cognate codon is difficult, or tRNA is difficult because the tRNA concentration is low, then that A site will be vacant more times than not. And then it allows for the fusion of the tRNA in the E site and the occupancy of that E site with NOT5 or C003. And then that leads to more events that basically allow for the mRNA degradation machinery to coalesce on the transcript. The details of those downstream events are something that we don't quite understand at this time. But one important point that I want to bring up is that the mRNA decay is a critical aspect in determining the overall proteome of the cell. So, such that if you look at a cell where we've depleted the protein that is responsible for monitoring codon optimality, and look at both mRNA levels and protein levels, you can see that you have a large effect on the overall expression of proteins according to their codon content, such that mRNAs that are enriched to non-optimal codons, those proteins will go up significantly in the absence of the C03, NOT5 regulator. And what's really important to notice in this graph is that even though I use the word non-optimal codons, that's really a word that we've ascribed to these types of codons. In fact, most genes within every genome use non-optimal codons. And so we really think about codons within the genetic code as either being codons or supercodons. And these supercodons, which stabilize mRNA, are restricted to only a small subset of genes. And these genes are ones that are highly expressed. They're like the glycolysis genes, ribosomal protein genes, etc. But non-optimal codons dominate the landscape of the genome. And by modulating mRNA stability, you can see you have a powerful effect on the architecture of the overall proteome. And this will be important for our next story. And so going back then, the mRNA degradation machinery is critical for the overall expression or levels, steady state levels of protein within the cell, and has a powerful effect on overall gene expression. So if we turn our attention now to this new growing field of mRNA therapeutics, since the COVID epidemic in 2019, we've learned that we can use mRNAs as powerful new drugs. And the utility of mRNA as a drug is expanding every day, with mRNA drugs now being used in treating infectious disease. These are vaccines, and there are many different viruses that we are now targeting with mRNA-based applications. But in addition to these, I guess you could call them now traditional applications, there are new emerging technologies that promise to use mRNAs for the treatment of monogenetic disorders, in metabolism and cardiovascular disease, as well as cancer. And so there's a lot of room to grow in this space. And one of the critical features that we have to begin to understand as we use this powerful technology is how do we improve the overall efficacy and durability of these mRNA-based drugs. And so just talking briefly about how we functionalize an mRNA to be used as a therapeutic. Essentially, we have plasmid that will encode your favorite mRNA. This gets transcribed in vitro, And critically, it must have at least 3 or 4 features. It has to have, if it's a linear RNA, and I'll just define these features as being critical for linear RNAs, it has to have linear mRNAs. It has to have a 5' cap structure, and this cap structure is critical for the translatability and stability of that mRNA. And then it must have a 5' untranslated region. It must have an open reading frame that starts with an AUG, and this encodes the polypeptide that you wish to express. It must have a 3' UTR, and then it has to have a polyadenosine tail, typically between 100 and 250 nucleotides. And then if it has these features, it will be loaded on the translational initiation apparatus, where the first step in translation is the recognition of this cap structure by a protein called EIF4E. And the presence of that unique modified cap structure, which has an M7G PPP and linkage to the 5' N, is what's needed for the initiation complex to assemble. And then the poly-A tail is thought to help this process by mediating the stability of the initiation complex. So what this tells us is that we actually can use if we're trying to improve the overall efficacy and durability of mRNA-based therapeutics, then the cap becomes an important area to focus our attention, because we in theory could improve the overall translatability and stability of mRNAs by changing caps. And I'm going to tell you a brief story about a novel cap analog developed by TriLink Biotechnologies that my lab has helped test the mechanism of action of how it works. And so this is a cap analog called M6, which has unique modifications present in these 2 positions that are not found in normal M7G caps. Now, importantly, work that TriLink has done, they've found that this M6 cap, in fact, outperforms other cap analogs, including their normal clean cap, which is this AG 3' methyl cap. And you can see in this mouse experiment that M6 is giving you a vastly improved translation of the mRNA that's been injected. And the quantitation of that is over here on the right. In addition, using a number of other metrics, you can see that the M6 cap is performing better than other cap analogs in every assay that has been tested. And I won't go into all the detail here. Suffices to say is that this cap analog, which was designed to improve the overall efficacy of mRNA-based therapeutics, is doing quite well. And so what my lab was really interested in is understanding the mechanism of how these modified caps, such as M6, could be improving the overall durability of an mRNA therapeutic. And so we focused on the 2 steps that the cap is important for: the binding of the translational initiation complex through the protein EIF4E, and the decapping of the cap by an enzyme called DCP2. And so just starting out, we purified human EIF4E. And again, this is the translational initiation protein that binds to caps and is required for initiation. And then we performed electrophoretic mobility assays to determine binding constants of EIF4E to cap, normal cap, as well as the M6 cap and just a triphosphate, 5' triphosphate labeled RNA substrate. And what you can see here is actually that the KD to a normal cap is about 72, but the M6 cap actually binds less efficiently to EIF4E, despite the fact that we're getting a much significant increase in efficacy in vivo. So it appears as though that M6 is not, in fact, binding more efficiently to the translational initiation factor EIF4E. In vitro experiments, we can then show that M6 has fairly negligible effects on the process of translational initiation. And so this is just an in vitro experiment monitoring translational initiation rate, looking at normal cap versus M6 cap, and you can see they're pretty much about the same. If anything, M6 is slightly reduced in its ability to promote translational initiation. And so what's going on here? And so then we turned our attention to the stability of the mRNA that has an M6 cap and developed a decapping assay The major enzyme that removes the cap in the human body is a protein called DCP2. We purified human DCP2 to homogeneity and then developed this in vitro decapping assay, which allows us to monitor how quickly an mRNA cap is removed. And essentially, this is based on a spectral shift assay. We're looking for a change in size and of the mRNA following removal of that cap. And not to belabor this, but essentially what you see is that if you look at the in vitro half-life or the decapping rate of the mRNA, when you have a normal cap, it's about 39 minutes. But when you have the M6 cap, you're in fact significantly inhibiting mRNA decapping and stabilizing that mRNA in vitro. And we can see that both in this assay as well as in vivo. This is an in vivo half-life where we've transiently transfected mRNAs with either a normal cap or M6 and monitored their half-life in vivo. And you can see significant gains in stability with the M6 cap. And so just to wrap this section up, the modification of the 5' cap such that it becomes resistant to mRNA decapping becomes a powerful mechanism in order to boost the overall efficacy of in vitro transcribed mRNAs. And just showing here a reiteration of everything that I've shown you, where different types of cap analogs have negligible effects on the overall abundance of the mRNA, but this M6 cap leads to a significant increase in the overall stability in vivo. Okay, so the conclusion I'll leave you from this part of the talk is that when trying to improve the overall efficacy of an mRNA therapeutic, the modulation of stability is a key feature that has to be considered, and that significant gains in the overall durability can be made by simply impacting the decapping rate of the mRNA. All right, so I'm going to turn now to a different story and talk about other aspects of translation and mRNA stability that we've been leveraging for therapeutics. And so I just told you a story about how we use our understanding of mRNA stability to improve the durability of mRNA-based therapeutics. But now I want to talk about other ways in which we can modulate mRNA metabolism using the knowledge that we have of stability and translation. And I'm really going to focus on dosage disorders or haploinsufficiency disorders. And these are a group of genetic phenomena where a mutation occurs in one of the two alleles. And this leads to a reduction in the amount of mRNA and protein within the body, or a reduction in the functional amount of protein, where there's half the amount of protein that's needed for appropriate metabolism, appropriate function. And so these haploinsufficiencies are not necessarily caused because of the genetic mutation per se, but simply because there is a reduction in the overall concentration of the final gene product. And so we've used our knowledge of mRNA stability and translation to try to develop novel technologies that in fact augment the expression of the wild type copy of the mRNA or the mutant copy of the mRNA in order to restore the haploinsufficiency back to a normal level. And so I'll first tell you about a story that started in my lab that led to the formation of Tevard Biosciences. And Tevard is a company that I formed in collaboration with Dr. Harvey Lodish at MIT and Daniel Fisher and Warren Lamert. And Daniel Fisher and Warren Lamert are both fathers whose daughters have a haploinsufficiency disorder called Dravet syndrome. And I'll talk a little bit more about Dravet in a second. But Dravet syndrome is a devastating epilepsy. And Tevard is Dravet spelled backwards. And the goal of Tevard Biosciences is in essence to turn the condition of Dravet around. And that's what we're hoping to do. So Tevard uses a novel technology called suppressor tRNA, and I'll tell you more about what these are in a second. But this is a platform technology that is really a one-size-fit-all cure for a broad range of diseases. And these are diseases that are caused by nonsense mutations. And so just to go back, nonsense mutations are mutations where there is a termination of mRNA translation because there's no cognate tRNA. And so what Tevart's technology, which is really a technology that dates back to the 60s, is to engineer tRNAs such that they read through stop codons. And so these are what are classically called suppressor tRNAs. And by reading the stop codon, it allows for the normal function of the normal protein to be produced from a nonsense containing allele. In addition to the suppressor tRNA platform, which I'll talk exclusively about today, Tevart has two additional technologies that I won't talk about, one of which is to use tRNAs to enhance the overall stability of an mRNA through this concept of codon optimality, which I introduced early on in the talk. But for today, we're going to concentrate on the suppressor tRNA technology. Again, just going back to the fundamentals of mRNA translation and the genetic code is composed of 64 words, and 61 of them encode amino acids. But 3 of those, UAA, UAG, and UGA, have no cognate tRNA that recognizes those 3 codons. And those codons instead are stop codons. They lead to the termination of the nascent polypeptide while it's on the ribosome. These 3 codons are in fact recognized by protein factors and not tRNAs. Importantly, however, up to about 22% of all human genetic disorders are caused by the presence of a stop codon prematurely within the gene. And that leads to the production of a truncated polypeptide that's non-functional. In particular, if we look at data from ClinVar and look at pathogenic nonsense mutations that have been filed in this database, 25 or about 23 to 24% of all nonsense mutations causing clinical significance, in fact, belong to the conversion of CGA arginine to UGA, and 20% are the conversion to TAG. And so this is really based on some simple properties of the spontaneous deamination of cytidine. But what this means is that with just a handful of these suppressor tRNAs, we can in fact correct a large number of nonsense mutations in disease states. And so the idea is really quite simple. We engineer a tRNA to an arginine, in this case tRNA, to recognize UGA rather than CGA. And when there's a premature termination codon, that tRNA will bind and now lead to the incorporation of arginine where it was supposed to be. And now the normal protein product is in fact made. This technology works quite beautifully. Here's an example of an experiment where we have a cell that's expressing the gene that's associated with Dravet syndrome. It's a gene called SCN1A. And you can see that we get normal expression here in this control. But if we express a allele of SCN1A that contains a stop codon at arginine 1407, you'll get no expression of this sodium channel gene. But if we transfect in the suppressor tRNA, we in fact restore the expression of that protein to near wild type levels. And these are controls using drugs that have been touted as nonsense suppressor drugs. And you can see there's really negligible effects of those drugs on read-through of the arginine stop codon. And just to show you that not only are we getting expression of the arginine of the stop codon containing SCN1A, this is also functional expression. So SCN1A is a sodium channel, and we can monitor the opening and closing of that sodium channel using a patch clamp assay. And you could see when you have the mutant SCN1A, you get no opening and closing, but when you express the suppressor, you now restore that to normal levels. The same exact suppressor can be used to treat multiple alleles. It's not necessarily relegated to one particular type of stop codon or one position. This is an experiment looking at patch clamp assays on SCN2A, but it's the exact same tRNA that I just showed you. But we're looking at multiple alleles here where the stop codon has been moved through SCN2A. And in each case, you can see the restoration of the opening and closing of the sodium channel by the exact same drug that I just showed you, but now in a new gene. In mouse models, we can extend the survival significantly of Dravet mice by expressing a suppressor tRNA. Typically, this haploinsufficiency model, these mice begin to die at about 20 days, such that by the 100 days, almost all of them have perished. But when we suppress multiple different types of suppressor tRNAs as AAV cocktails, that we get restoration of, we extend survival of these mice significantly. In addition, we can verify that we have restored mRNA levels when we express the suppressors, and we also can reduce seizure, heat-induced seizure tolerance by significant numbers of degrees expressing these suppressors. And then lastly, the amount of protein is rescued up to 76% of wild type. And so we're already seeing very nice data that these tRNA-based suppressors can be used to treat a devastating haploinsufficiency like Dravet syndrome. But what's important to think about is that I had mentioned that almost 22% or so of all genetic disorders are caused by a nonsense mutation, and most of those are an arginine CGA going to UGA. So what that means is that this gene therapy modality has the potential to be a one-size-fits-all for multiple indications. And here's an example of how we can, in fact, utilize that concept. These are a number of haploinsufficiencies associated with the CNS, including Dravet syndrome, which is SCN1A, but other indications that are not related to Dravet syndrome, but also cause Dravet-like phenotypes. But as you can see, that if you look at the frequency of arginine to UGA mutations in each of these indications, it's actually quite high. And we can, in fact, use the exact same drug to treat multiple indications by the same route of administration. And so that allows us to think about this gene therapy technology as a platform technology that can be used in a number of ways. And here's an example of that, again, done in mice, where we're using the exact same drug that we showed for Dravet, but this time for Rett syndrome, and looking at the expression of the protein associated with Rett syndrome, MECP2. And as we add high doses of the suppressor, you get restoration of MECP2 expression in this mouse model. And we've seen similar results in mice where a mouse model for CTKL5 deficiency. And so we really think about this tRNA suppressor platform as having broad applications for these rare genetic disorders associated with nonsense mutation. At Teva, we continue to optimize our tRNA candidates in a number of ways. One of the ways we do that is by high throughput screens of suppressors, as well as by using and optimizing based on the principles of evolution of tRNA sequences. And then we're also employing advanced machine learning and artificial intelligence in order to select for novel tRNA structures that improve the overall efficacy of suppressors. And so what I've shown you then is that we can use a tRNA-based approach as a way to go after haploinsufficiencies, such as Dravet syndrome, Rett syndrome, and other devastating indications. But my lab wanted to go beyond some of the technology that we developed at Teva and ask, are there other ways that we could, in fact, improve gene expression at the level of mRNA stability in a transient fashion? And so we go back now to thinking about mRNA turnover and what controls mRNA turnover, and I've shown this slide a number of times. And as I mentioned at the beginning of the talk, a key determinant in monitoring mRNA decay rate and mRNA translation is, in fact, the poly-A tail. And the poly-A tail is removed at a constant rate, and that rate is variable between each individual mRNA. It's fast if that mRNA is unstable. It's slow if that mRNA is stable. And the technology that we have developed is, in fact, a technology to fool the mRNA into thinking that it still has a poly-A tail. As long as an mRNA has a poly-A tail, it will translate. And so what we've created are small oligonucleotides that bind to the 3' untranslated region of the mRNA and have a poly-A tail mimetic. And essentially, by having this poly-A tail mimetic bound to the 3' end, it stimulates the translation of that mRNA and keeps it going longer than it normally would in nature. And so we call this technology mRNA boosters, and it's a novel therapeutic candidate for the treatment of a large number of haploinsufficiency disorders. These are very small oligos, and so it allows us to do a lot of chemistry on these. But basically, the anatomy of this is as follows. There's an antisense guide region that's complementary to a region within the 3' untranslated region of an mRNA. We have a poly-A tail of a discrete size, and then we have modifications peppered throughout the transcript in order to improve the overall durability and efficacy. In our first set of experiments that we tested this technology, we simply took RNA oligos that were complementary to specific mRNAs, and we polyadenylated them in vitro using poly-A polymerase. And this was a really straightforward way to screen through large numbers of RNA oligos in order to identify candidates that would give an enhanced effect on mRNA stability and translation. In the end, what we get are oligonucleotides that are polyadenylated between 100 and 300 nucleotides. One of the first experiments we did was to simply co-transfect a booster RNA with a reporter that has a binding site within the 3' UTR. And so this is a in vitro transcribed luciferase and a booster that is binding to the 3' end. And in addition, we co-transfected in Ranilla as a control. And what you can see here is that when that booster RNA does not have a poly-A tail, there's no stimulation of translation of Firefly versus Ranilla. A10, nothing really happens. But when we have an A200 tail, we get about a 6-fold effect on the overall translatability of that mRNA, irrespective of the ratio of the booster to mRNA that we've transfected. And so this was positive preliminary data to suggest that this technology was going to work in human cells. And then we moved on to test whether or not we could augment the expression of an endogenous mRNA in vitro. So we have, over the years, studied this little protein called LSM8, or we've studied the mRNA. It's a very well-behaved mRNA that we know a lot about. And so our first, although it's not clinically significant, our first experiment was to develop an mRNA booster directed against the endogenous LSM8, and then transfect that booster at various concentrations into human cells. And this is a representative experiment where you can see that by even 20 nanomolar of the booster, we've already saturated out the effect, and we're seeing a 3-fold stimulation in the expression of that particular endogenous mRNA. Now, LSM8, as I mentioned, is not a clinically relevant mRNA, so we decided to turn our attention to mRNAs that are involved and associated with haploinsufficiency disorders. And so we developed boosters against a variety of these, including MECP2, which is linked to Rett syndrome, the beta-catenin mRNA, CTNNB1, linked to beta-catenin deficiency syndrome, pure alpha, also another haploinsufficiency syndrome, and SYNGAP1 deficiency, which is also a haploinsufficiency. I should point out that most of these that I'm talking about here are actually haploinsufficiency disorders that predominantly affect the CNS and are in the epilepsy autism spectrum disorder type indications. And what you can see is that in each case, in culture, we can develop a booster that has a significant effect on the expression of those endogenous mRNAs, such that we can see between 2 and 4-fold effects depending on the particular mRNA. We then expanded this to ask if we can recapitulate these effects in vivo. Here again is the MECP2 booster. We're enhancing not only its mRNA expression, but its protein expression in a dose-dependent manner. But then we've encapsulated these RNA boosters into LNPs and done tail injections into mice. And then you can see that when we harvest the liver, these particular boosters, B2 here, are having a nice effect on both the mRNA expression as well as the protein expression of that particular gene in the liver. Now, MECP2 is a X-linked gene, so it may be difficult to treat with this type of with this type of therapy, but nonetheless, it provides a good proof positive that, in fact, we can use this technology in, at least in mice, to improve the overall gene expression that we observe. In addition, we've shown similar results is work that we've done in collaboration with Michelle Jacobs at Tufts University, where we focused on beta-catenin mRNA. And again, as I showed you a few minutes ago, we can get boosters that enhance the expression of the beta-catenin mRNA, as well as its protein expression. And this is, again, showing in a dose-dependent manner that we can improve the overall expression of the beta-catenin protein. And in a in a tail vein injection experiment, we can improve the expression of beta-catenin within the liver of these mice. And so not only do we see this with MECP2, we see it with a second gene of interest. Beta-catenin is part of the Wnt signaling pathway. And so one important control is to determine whether when we enhance beta-catenin levels, are we getting engagement of downstream transcriptional targets? And in fact, you can see that we are. And so this is an experiment where we've introduced our booster to cultured human neurons, and you can improve the expression of beta-catenin in these cultured neurons, and that also leads into a concomitant increase in a downstream Wnt target, EN2. And in similar experiments we've done in cell culture, in HEK cells, we can also see an increase in other known markers of Wnt signaling, including c-Myc and DKK1. And so this demonstrates to us that not only are we increasing the expression of beta-catenin, we're increasing the functional output of the beta-catenin gene and getting target engagement of downstream genes. And so then lastly, we've focused most of our effort on developing technology toward SYNGAP deficiency. SYNGAP deficiency is a serious debilitating monogenetic encephalopathy. It does have phenotypes that are fairly similar to Dravet syndrome, Rett syndrome, CDKL5 deficiency, and Angelman. Of course, there are differences in all of these autism spectrum disorders, but it is a devastating disease of the CNS. It affects about 1,700 patients worldwide. SYNGAP1 is an important gene required for normal neuronal function and present on chromosome 6. And like many of these autism spectrum disorder genes, the number of mutations that you see within the human population is sporadic. So these are often the result of germline sporadic mutations, and they're not necessarily the result of inheritance of a founder effect. And so in terms of developing gene therapies that can go after these patients and help ameliorate their condition, it becomes challenging because there are so many unique variants that we see. This is true of Dravet as well as SYNGAP and many other encephalopathies. So technologies that are mutation agnostic become sort of the favorable way to think about treatment of these encephalopathies, because we don't necessarily have to concern ourselves with the type of mutation present in the diseased allele. In addition, we're challenged by, SYNGAP is like many human mRNAs, we're challenged by the fact that there are multiple spliced isoforms. There are at least four known splice isoforms for SYNGAP1, and it's clear that the translation of all four is in fact critical for appropriate gene function. And so by using technologies that might improve one of these isoforms over another, that doesn't necessarily provide us with the opportunity to fully improve the overall normal expression of the SYNGAP1 protein. But one thing I will note here is that despite the fact that there are multiple splice isoforms, and this is true of many alternative isoforms in the genome, they share a common 3' untranslated region. And so our booster technology, which recognizes the 3' UTR, will in fact bind to all four splice isoforms and presumably lead to a restoration of each of their protein function. So as a preliminary experiment, we developed booster RNAs that work in neurons, and you can see that we have boosters that work between 2 to 4-fold effects on SYNGAP expression. And then the gone about trying to determine regions within 3' untranslated region that would be best to target for this particular indication. And of interest is what we find is, despite the fact that 3' UTRs are relatively long and highly conserved between mice and human, we see that there are particular regions that, when targeted, have better effects on the overall expression of SYNGAP1 versus others. And my lab is currently trying to figure out why this is true, but it may have to do with a lot of things, such as access to the actual 3' UTR, not competing with other RNA binding proteins or RNA structure, or may even reflect a natural need for boosters to be in particular regions relative to the termini of the transcript, but we don't know right now. Anyway, using this type of approach, we've been able to select lead candidates for further testing, and then we've moved on to take those boosters and inject them directly into the hippocampus of mice using a technology developed by a colleague of ours over at the Homewood campus in Johns Hopkins. And in this case, what we're doing is we're injecting into the hippocampus a superelastic tube that, when retract, will leave behind a mesh that we can use to then perfuse small molecules into the brain directly over time. And using that approach in a preliminary experiment, we perfused SYNGAP boosters directly into the hippocampus. And as you can see in this experiment, where we have 3 mice, the control has essentially a normal amount of SYNGAP expression, but we get about a 2 to 3-fold effect on the protein expression in the hippocampus, and that permeates out into other regions of the brain, including the cerebral cortex and the midbrain. But by the time we get to the cerebellum, there's really not much SYNGAP expression there anyway, and maybe there's a little bit of an effect in the cerebellum, but it's negligible. And so this was an exciting observation for us, suggesting that we can in fact get these booster transcripts to augment the expression of an endogenous mRNA within the brain of clinical significance. We've repeated this experiment with more mice, and again, this time only focusing on the hippocampus, and showing that in a variety of mice, male and female, you can in fact see a significant stimulation of SYNGAP1 protein expression in the hippocampus. And then lastly, in ongoing work within the lab, we're in the process of modifying these boosters to improve their overall durability. As I mentioned, they're small, so this allows us opportunities to do unique chemistry. And we've gone about this just starting by incorporating known modifications that improve the overall durability of these transcripts, including phosphorothioates and 2' methyl groups. And in just preliminary experiments, I can show you that, in fact, these still work, and they work quite well. And we improve the overall durability over several days at this point. So we're optimistic that we can continue to play with the chemistry of these and improve their overall durability, such that they could move into good clinical candidates. So just to wrap up, what I've shown you then in this last part of the talk is we've developed a novel technology that can augment the expression of mRNAs in mice, and we've shown this on the MECP2 mRNA and the beta-catenin mRNA in the liver, and then more recently shown that we can augment expression of the SYNGAP1 mRNA in the mouse hippocampus. And so we believe the mRNA booster technology is a novel gene therapy that enhances protein expression. It's really a first-in-class technology that leverages the power of RNA therapeutics. We believe it will be disease modifying in that it restores the normal level of the protein back to wild type. We also believe that this will be mutation agnostic, because we can specifically affect the wild type mRNA. And we have shown it's really a platform technology, because we can use this across multiple indications. I should point out that in the human body, almost all mRNAs, with the exception of a small number, have a poly A tail, and that poly A tail is critical for driving translation instability. And so there's no a priori reason why this booster technology could not be used on the vast majority of mRNAs within the cell. And then just in the last 2-3 minutes, I want to leave a plug for some of the work that we're doing in the mRNA therapeutic space. I'm one of the founders, and Johns Hopkins is one of the founders of what's called the Alliance for mRNA Medicines, or AMM. I'd invite you to look at this website if you want to learn more about the Alliance. But essentially what the Alliance is doing is we're very interested in moving forward the mRNA therapeutic space because we recognize the power of this type of approach to benefit patients as well as public health and society. And so our mission is, in fact, to help advance this technology to the next level, as well as improve patient lives and advance the scientific knowledge by empowering mRNA industry leaders, innovators, and scientists, as well as other key stakeholders in the industry. And so the AMM has brought together well over, this is a list that's growing almost every day, over 50 industrial partners that are focused on mRNA-based therapeutics to come together to harmonize around conversations in this space. And so, as I said, if you're interested in this, please reach out to us and consider becoming a member. Some of our key activities will be it's important to understand that we have to combat some misinformation out there associated with mRNAs and mRNA vaccines. We also want to become advisors to key regulatory agencies, policymakers within the government, as well as stakeholders within North America, Europe, and the Asian peninsulas, as well as become a source of reliable mRNA information for the sector and provide an opportunity to convene the community and amplify our information to scientists and other organizations. And so with that, I'll just conclude and first thank the members at Tevard Biosciences. This is the work I talked about is done exclusively in those labs at Cambridge. And this picture is outdated. We're now about 50 full-time employees. And if you're curious of what we do, please go to our website. And then thank the people in my lab who have done this work. The work that we've done in collaboration with TriLink was done by a postdoc in my lab, Zach Mandel, who's shown here. And then the work on mRNA boosters was done by Bahare, who's shown here. And thank our collaborators as well as funding sources. And thank you for your attention.
If you have a question for our speaker, please input your questions in our Q&A box and stay tuned for our live Q&A session at the end of the webinar. With that, let's bring Dr. Jeff Coller to the stage. Thank you, Dr. Jeff Coller, for that wonderful presentation, and welcome to our live Q&A session. If you're in the audience, please feel free to enter your questions in the Q&A box. So our first question says, how do the suppressor tRNAs not connect to non-premature stop codons or not normal stop codons that terminate the normal protein?
Yeah, that's a good question, and it's a question we get asked a lot. So the best answer we can give is that if you think about this, the cell has a way to discriminate normal termination codons from premature termination codons. Easiest way to think about this is the existence of nonsense-mediated mRNA decay, where a premature termination codon will elicit NMD, where a normal termination codon does not. And so that tells you automatically, although not well understood, that the cell can discriminate between normal and premature termination codons. And then in experiments that we've done with tRNA suppressors, we see no read-through into the 3' UTRs of any mRNA when we overexpress any of the suppressors against the 3 various stop codons. In addition, we've done very deep proteomic analysis on mouse tissue after expressing a tRNA suppressor, and again, see no novel read-through of normal termination codons. So it suggests that there's a normal process existing in the cell that discriminates between normal and premature. So it works very well for us with the usage of these tRNA suppressors.
Have you tried to do mRNA boosters of different lengths? How critical is the length of the poly A?
Yeah, so the length of the poly A tail is something we're playing with right now, and to reduce the size, we don't really see much of a difference between, say, a 300 nucleotide tail versus 100. We're presently reducing that to see what is the minimal binding site we would need for poly A binding protein. In theory, poly A binding protein binds to 12 A's but protects 25. So for a single molecule of poly A binding protein. So it may be sufficient to have 25 to 50 A's, but those experiments are ongoing right now.
What are your thoughts on the efficacy of predicting and modifying RNA secondary structure in order to increase half-life and protein expression?
Yeah, I guess it depends on where you're talking about, because if you're referring to IVT-derived mRNAs, the rules that govern stability and efficacy or stability and translation of a exogenously delivered mRNA are different than those that regulate normal mRNAs. So structure does seem to impact the overall stability or efficacy of an IVT-derived exogenous mRNA. And there's a paper that came out from Melissa Moore a couple of years ago in PNAS, in fact, documented this. But the influence of structure and such on decay of normal mRNAs doesn't seem to be as critical as it does with IVT-derived mRNAs. And that may have to do with, essentially hiding the mRNA from the innate immune response. So I would argue that we don't know all of the rules that dictate the stability and translatability of IVT-derived mRNAs, but we're learning those as we go along.
Does the booster anchor need to be with the 3' UTR? Could a booster work if it bound to the 5' UTR?
It's a good question. I mean, we put it in the 3' UTR for a variety of reasons. One is that that's where the poly A tail is. So it's sort of a natural place to anchor an exogenous intrans poly A tail. There's no, as far as I know, a priori reason why it may not work in the 5' UTR, but we haven't tested it. One of the advantages of tethering it to the 3' UTR is we have a lot more sequence space to choose from, because 3' UTRs tend to be fairly large. And we, in general, do not see a position-dependent effect for if we can get boosters that will enhance mRNAs regardless of where we put them in the 3' UTRs. Some sites work better than others, but we haven't rigorously tested whether 5' UTRs or other regions of the mRNA could be accessed and stimulated by the technology.
How do you get mRNA-specific boosters just based on the hybridized region, and how long are those regions?
Well, the specificity comes from the nucleotide sequence, and we select nucleotide sequences that are, of course, unique to those particular transcripts. And in general, our guide sequences are about 30 to 50 nucleotides in size.
Do you use any frame shifting based technology?
No, we don't. And the tRNA suppressors that we use in Tevard, we don't see any enhanced frame shifting associated with them. So that doesn't appear to be an issue for us.
Great. We're just about to wrap up the Q&A, but maybe one or two questions to squeeze in. What is the delivery method of the engineered tRNAs to dose in vivo?
Yeah, so we deliver the tRNAs through AAV vectors, and the choice in that was deliberate because there are a lot of unique modifications found in tRNAs. There's about 150 unique modifications that could be found in tRNA populations. And we wanted to allow the cell to add those modifications rather than do it in vitro. The other reason for this is durability. We're looking at these ultra-rare genetic disorders, specifically of the CNS, and we wanted one-and-done type approaches rather than a way to dose over the lifetime of the patient, which is not necessarily practical when you're dealing with a CNS disorder. So AAV was the right choice for us in this particular case.
Great. And we're just at the hour and wrapping up our Q&A session. Thank you, Dr. Jeff Coller, for joining us and that wonderful talk. Thank you, TriLink, for supporting this webinar, and make sure to catch this webinar on demand, which will be available in the next 48 hours. So until then, we'll see you next time. Thank you for joining us.
Thank you.